Hello and welcome to the Deep Sea Podcast, a fun take on a science podcast about everything deep sea. I'm Dr. Thom, curator of fishes at the National Museum of New Zealand, Te Papa Tongarewa, and with me as ever is Professor Alan Jameson, Director of the Deep Sea Research Centre at the University of Western Australia and scientist for Inkfish. Any of our dumb opinions are our own and do not reflect the opinions of our respective organisations. Hello, mate.
Hello.
Hello. How are you coping in the heat?
It's not the heat that's getting me down today. It's just come off the back of a 5-day conference with 50 deep sea people I've been organising this week, and I'm exhausted. And Friday night was a bit wild. So it's now Saturday evening and I still feel rough. There was lots of friends of the podcast there. Georgia was presenting. Old mate was there. He cooked a barbecue for 50 people. Heather Stewart was there. She's been on the podcast before.
That's an Aussie wedding, isn't it? Cooking a barbecue for 50 people?
Yeah, pretty much, yeah. Did it on the shores of Matilda Bay.
Oh.
It's very good. He did well. He did very well. As you would expect. They called it Old Mate's classic Aussie sausage sizzle. Yeah, very good. Oh, Jakob was there as well, of course.
Yay. Now celebrity Jakob. Folk like that episode. It was really nice.
That was great. I was listening to it in the car driving around the Canary Islands a month ago. That's quite funny.
It's a great story.
Yeah.
His sort of observations and bringing it full circle. It was really enjoyable. Anyone who didn't catch that episode, that was our eco stop where Jakob talks about growing up in a culture where there wasn't really a concept of the ocean and then finding himself at 5,000 meters deep. Yeah, really nice story.
Yes. It was a good week. Lots of good talks, lots of geology, biology, ecology, oceanography. Big mix of everything we've done in the last few years and good chats about what we're going to do in the next 5 years. Really good. I'm just exhausted. It's really hard work organising a science conference. It's hard. Oh, and Devin was there as well, our glaciologist from Antarctica. He was there.
I feel rough and tired as well, but my reason maybe isn't quite so good. I got up at 2:30 in the morning to join some local fishers who had kindly offered to take me and Kat out to deep water to test some of our new equipment before we head off to Antarctica again. So I just want to say a massive thank you to William and Richie from W. McWilliam Fishing for taking us out. And I'm sorry I was sick the whole time. I was really spewing. But these guys send some crab pots down really deep into almost 1,000 meters. Richie has an Instagram where he posts some of the most interesting stuff that they bring up, and they return everything that they can. So everything that's alive, they return. So you can check out tailsfrom_the_deep on Instagram to see some of those pictures. It's a really nice blobfish as well. And apparently the blobfish was fine. They released it and it went straight back down. So thanks for the help, lads. Really appreciate that today. We wouldn't have been able to do that otherwise. My other bit of news is I've got an exhibition opening soon. It's my first time curating in a museum-y way. And I did a curate. There isn't a single fish in it, but it does explore sort of our relationship and place within the natural world. It turns science into art, which is why I got involved with it. So it's a London-based collective called Marshmallow Lazer Feast. They do things like LiDAR scan trees in the Amazon, but then also scan below the soil surface and create these incredible digital arts where like you can see the nutrients coming up the trunk and things like that. And it's all about sort of breath, the tree exhaling as you inhale kind of thing. It's really nice. It's quite spiritual, but it's sort of grounded in science. So that's going to be in Te Papa in a couple of weeks, all over the summer. It's called Breathe Mauri Ora. And so anyone in Wellington, please come and check it out. And we've got some events coming on as well, so you might see me doing things related to the show. But I've not done that before. That was really nice, actually. I got really into the show, and nice palate cleanser. Like I say, there's not a single fish in it, but it dealt with some quite big concepts that I quite liked. So anyone who particularly liked our eco-anxiety episode, touches on a lot of those feelings, really, sort of being connected to and part of nature. Really good fun.
Are people expected to part with the hard-earned money to come and see you doing a curate?
Yes.
Oof.
Yes.
Oh, that's a hard sell.
My family, and in particular my son, is very upset by the fact that people pay money to hear Daddy talk, but Daddy talks too much and he is boring. But some people like it.
Maybe he'll grow up to start a business where people pay him to stop you talking.
He just comes into your house and deletes the podcast.
Yes.
You don't need that. You don't need that, man. I lived with him.
Yeah.
It really gets grating after a while. We couldn't do the show without our patrons. It really is what keeps it going. So we just wanted to say a massive thank you this month to Elizabeth Grace Demir and Ness Morgan. So thank you to our new patrons who keep the show going. We've not done a song of the month in a while, and I gotta say that's because I'm afraid of Spotify. They've gotten really fastidious with the tagging of any music in podcasts. Annoyingly, there's no human beings that review that. So just a robot notices that there's a piece of a copyrighted piece of music on there and then takes your whole show down. And it's like a week-long appeals process to try and get a human being to then reinstate it. And that can be quite damaging to a podcast if you disappear for a while and then sort of come back. So yeah, I've been a little bit nervous, but I like a bit of music. I like the little segue. You had one on the Discord, Alan. There's something, you saw them live. You gazed upon their visage.
Yes, when I was in the UK recently, I went to see Megadeth, took my son to his first metal concert. Megadeth, Support and Disturbed, which I don't agree with that order of events, but I like both bands, but Megadeth should never be a support band. But they were great, absolutely brilliant. So actually, I could send you a video with audio of said son playing Megadeth.
Oh, perfect.
Yeah.
Okay.
Spotify's not going to get you there.
No, that's original content. Unless he's really, really good and it sounds like the album version.
He's nearly that good, yeah. Well, we'll see. We'll let the viewers decide.
We'll let Spotify algorithm decide.
Yes.
Grant, do you want to introduce it? What's the track?
He's learning to play a song from the Rust in Peace album called Tornado of Souls.
Nice. By the way, we've got a lovely new website up and running at www.deepseapod.com. So that's where you can find loads more information about any of these news stories. We always try and put some wider reading on there, lead you to the papers or the larger articles or videos. So if you ever want to know more about these things, just hop on over to there.
So in the news this month, the world's largest rays may be diving to extreme depths to build mental maps of vast oceans. So this is a team of scientists from Peru, Indonesia, and New Zealand who've been tagging manta rays around New Zealand, and they're diving as deep as 1,200 meters. Which I think the more people tag, the more these depth ranges are getting bigger and bigger and bigger, right? I think things go much deeper than we think they do. So these deep dives are typically followed by extensive horizontal relocations, which kind of suggest that the dives are helping the rays gather navigational information about the environment.
Like we would pop our head up out the water to check where land is and then pop back down and swim. They're doing that the other way around.
Diving deep, coming up, moving along a bit, diving deep again, and spending very, very little to no time at all at depth, which means they're not doing anything on the bottom.
It might be a good way of picking up the magnetic field or maybe feeling temperature changes, so feeling deeper current directions. Maybe surface currents change quite a lot, but if you can get deep enough to those dominant currents, it's quite a good way of like, right, that one's heading north.
Yeah, it's pretty cool, huh?
Yeah, they're up to something.
A manta ray over 1,000 meters. That's going to mess up George's eDNA, isn't it?
I bet they're leaking like a sieve. I bet there's loads of DNAs coming off them.
Yeah, but heaps of DNA being squeezed out of those rays.
Yeah, it's a big plate with a face.
Yeah, it's got a weird face as well.
Scoopy face.
Yeah.
It's a new AI tool for deep sea research. So it's a deep sea exploration artificial intelligence model named Depth GPT. And it was developed by a team led by a Chinese scientist and was recently launched to help explore the deep sea. Interestingly, the model combines AI technologies such as deep learning, large language models, computer vision, knowledge reasoning to analyze various types of data, including video footage, topography, hydrodynamics, sediment, and bioacoustics. So there's a few things that have gone along this path, mainly sort of image annotation stuff, but this sounds like it ingests lots of different types of data and tries to sort of harmonize them. I'd certainly like to take this for a spin and see how it works.
Do it.
Yeah. I'll just ask to borrow it, mess with it.
Yeah. Just get stuck in, Tom. Just get stuck in.
Okay, see if I can break it.
And remember, and before you get stuck in, remember, whisper the words for science.
Smash. Depth GPT is a product of the Digital Depth Project, which is dedicated to studying deep sea ecosystems and was launched as part of the UN Decade of Ocean Sciences for Sustainable Development. So fun new tool coming out, big data, lots of data going in, revealing patterns that might not be obvious to the human observer, things happening on a large scale, and interesting correlations between things. What's going on with fat molecules, Alan?
I don't know.
A fat molecule's doing something, maybe in mud.
Are the molecules made of fat or particularly chubby looking molecules?
Oh, little chunky molecules.
Yeah, just little rotund molecules. Yeah, I mean, fat molecules been found in deep sea mud volcanoes somewhere around the Mariana Trench by the good German ship Sonne. Sonne. We love the Sonne. On a side note, I happened to be on a river cruise yesterday with my 50 delegates on a 2-story high glass cube that goes up and down the Swan River, and one of the songs came on was the final countdown. I'm not going to put any context for the viewers there.
Nope. Just for
But,
me. Just for me and Al.
Thom plus Zona plus Spinal Countdown is a great story.
Maybe kazoos.
Anyway,
Yeah.
yes, the fat molecules found in a mud volcano near the Mariana Trench. So locating life in these bright blue mud samples wasn't easy, apparently. The sparse number of cells identified and the low biomass pulled from the samples made it impossible to identify species by analysing their DNA. Cut a long story short, the microbes are archaea, not bacteria, and they're not viruses. They're just another thing, but small. Remember I was saying before, like, I don't understand anything I can't drill a hole through.
Yeah.
Archaea are one of those things. Yeah, they're in that,
Put it on the
top
list.
drawer of things I'm putting away because I just can't get my head around them at all. They're tiny, even if they're fat.
They're still tiny, even the fat ones.
Yeah, they're just fatter. Anyway, the team also examined carbon isotopes from the sample and estimated that various groups of archaea have existed in the mud volcanoes over time. Earlier dated bacteria combined resources to produce methane. However, later living species used sulfates and methane oxidization to survive. So I guess that's a big deal for people who like small things.
It sounds like they're doing something important.
I think sometimes I should spend more time going and reading the source material before we read out these news stories.
Putative promiscuous symbiont in deep sea corals and crinoids. After our chat last month about some very involved symbionts, it turns out they're not all that committed, that monogamous. So a new study has indicated that there's a potential cross-host microbial interactions between deep-sea corals. I know Desmophyllum, Pertusum, but Solenosimula variobalis.
Well done on the pronunciation there, Thom. You nailed it.
Anyway, corals and their associated crinoid. So basically, a couple of types of coral, 2 different species of coral, has an associated crinoid, those basket stars that lives on them. So anyone watching the ROV dives will see quite often lovely branchy coral, weird tangly up starfish living on it. So it's those 2. This was a study in Campos Basin in Brazil, and the crinoids, the feather stars, the basket stars are frequently found in association with these corals, and both corals and crinoids host symbiotic microorganisms like most things do, as we learned last month. Yet the physiological and microbial interactions between these organisms and the functional roles of these symbionts, particularly in deep sea environments, are largely unexplored. That's why I had to go and find some shallow squid last month. Using multiple approaches, researchers have found that the corals and crinoids both host the same symbiotic microorganisms, and metagenomic analysis revealed one of these symbionts contains a genetic pathway capable of reducing nitrate to ammonia, which could provide a substrate for ammonia oxidizing archaea.
Archaea.
Archaea again. It's Archaea
Yeah.
month. The study suggests that nitrogen cycling plays a key role in structuring microbial symbiosis in deep sea coral crinoid and metabolic interactions like DNRA-driven ammonia provisioning, which could underpin resilience in nutrient-limited environments. So it seems that these corals with their associated echinoderm buddies that we always see grabbing onto them have very similar microbiomes, and that these microbiomes include pathways that might benefit them by reducing nitrate to ammonia and things like that, things that they can get nitrogenous goodies from, in nutrient-poor environments. Well, fair enough then. In another news item, fresh water under my deep sea bed. Rather than trying to figure this out myself, I will throw to the scientists themselves.
Did you know that there is fresh water beneath the ocean? My name is Rebecca Robinson, and I am an oceanographer from the University of Rhode Island. I was recently lucky enough to participate in a multinational expedition to study a large deposit of freshened groundwater beneath the ocean offshore New England. The expedition included scientists, technicians, drilling and ship's crews, all united in an effort to retrieve freshened groundwater. The 74-day expedition was sponsored by the U.S. National Science Foundation in collaboration with the International Ocean Drilling Program, IODP Cubed, a European and Japanese partnership focused on scientific ocean drilling. We took cores and sampled waters from 3 sites offshore Nantucket. We cored over 800 meters of sediment and sampled over 10,000 liters of water. Typically, the water that sits between the grains of sediment that make up the seafloor look a lot like seawater, with a salinity close to seawater, which averages 35. But we found salinities of 8 or less, almost fresh enough to drink. The samples will be used to to measure the water's chemical composition, examine what types of microorganisms live in this brackish water, and to evaluate how it got there and how old it is. Humanity is thirsty for fresh water, and reserves like the one we sampled are found worldwide, making this study an important early step towards understanding whether this water is potentially useful for human activities. To learn more, go to ecord.org/expedition501.
So, how are you going to go from that to what the subject of this podcast is?
We've been spoiled this month, basically. Last month, it was the CIAC Symposium or Kayak Symposium. So it's the Cephalopod International Advisory Council, also known as Kayak Symposium. And this year it was in Japan. And all of the cool tentacly kids, including Kat Bolstad and the Squid Squad, attended. And Kat was very kind and was a roving reporter for us and accosted some of the most interesting tentacly, slippery, suction cuppy people she could find and talk to them about their work, including some celebs, actually. I'm quite excited. Thanks for coming on, Associate Professor Kat. Wait, there's a track changes in this document, Kat. The associate's been crossed out. What's that about?
Yeah, the associate's been crossed out. Well, apparently I'm allowed to profess now, and I'm going to flex my new professing muscles by hijacking your podcast once again for yet another Cephalopod special. My real goal for this is just to do it so many times that you stop saying Cephalopod.
Oh, sorry. I actually flip a lot. Is it Ceph?
I know.
But in Latin, isn't it a K sound?
Well, who would know? Who's alive?
No
who
one was
actually
alive.
speaks Latin? Quo plus in cana.
You went along to the conference. All the cool kids were there in tentacles.
Yeah, so this is the triennial or every 3 years Cephalopod International Advisory Council Symposium or Kayak Symposium. This year it was in Okinawa, Japan at the Okinawa Institute of Science and Technology. We had about 250 delegates, which I think is the largest one we've ever had. The previous one was a hybrid online in Portugal during COVID, and that was the first time we'd ever had it hybrid. It was really great to be able to participate from New Zealand because we were in lockdown, but it really wasn't the same as being there and catching up with everybody. Yeah, over 200 toothologists or squidologists, as we sometimes have to say, otherwise people think we're dentists, and quite a lot of extra talent thrown in there. So scientists of all career stages, but some of those are also excellent science communicators, authors. So we had Dana Staff selling some of her new cephalopod books, written a number of excellent cephalopod books. We had artists and photographers and filmographers share their really beautiful footage of these animals alive in black water and some deep sea footage as well. And it was a really great meeting. We covered all sorts of interesting topics, and some of them were deep sea. And so I shoulder tapped a few people to either have a chat with me or to record some little bits about their research themselves, which I thought I'd share.
Thank you so much. This episode, I have a feeling, is going to run long. We might wrangle it a little more for the pressurized version, but I think these are such cool people to hear from. This is such like bullet point rapid fire of lots of different topics. I'm just going to let it run long. So anyone listening in, just settle in. You've probably seen the counter on your podcast app, so you know what you're in for. But I think this is all gold. So I'm just going to let it breathe. I'm not going to part to it or anything. And if you want to get the punchier version, we'll try for it in the pressurized version. But to be honest, this is all gold, and I'm just going to let it run long. But when you were mentioning about the last one being a virtual conference and how much better it is to be in the room, you got lots of fun stuff in. Like you were diving and going bug hunting with some really cool people. Like that's why this social and the network of a conference is so nice.
There's the stuff you learn from people's talks. There's the stuff you hear people are doing, and then you have chats, and maybe you start a new project with somebody, or you talk to someone who's interested in coming and working in your lab for a bit. But then also you do things like you go off and do social events. I'd been in Japan for a couple weeks already, and I will say Japan was excellent bug hunting on my own, but turned out that there was a fair group of us who were really interested in the bugs of the area. We were in Okinawa was quite tropical, so there were some very good, very large bugs around. Meg Midland, who we'll have a chat to, was a particular spider fan and hadn't seen any spiders in Japan yet. So I made a point of relocating a large one out of the women's bathroom in my drink bottle to hand to her so that she got to see at least one large spider.
That's friendship. I brought you a spider.
I brought you a spider in my drink bottle. I will share the photo. I have a photo of her with it on her hand, and she looks so excited. And we did go diving. There were some live cephalopod encounters. So I got to see my first wild cuttlefish ever. And I got to shake hands with a new species of octopus that I hadn't encountered before. It was a little abdopus. So other people also saw bobtail squids and some other cuttlefish as well. So there was an excellent array of cephalopod exposure at this meeting.
Okay, let's dive in. Who are we going to talk to first?
Leading up to the conference, there's always a couple of days of workshops. We had workshops about fisheries and about AI for cephalopod science and how that might move us forward. The one I participated in was about cryptic biodiversity, because there are still many, many cephalopod species left to be described. And quite a lot of those are things that historically people have sort of said, oh, worldwide, I think it's one species. And then the more you look, the more it's not. So this workshop was really summarizing our current state of knowledge about how much we have left to discover in the cephalopod biodiversity space. And so I had a chat to Fernando Angel Fernandez Alvarez, better known as Fafa, who was organizing this workshop and is one of the other leading deep sea squid biodiversity people worldwide.
Hello, everybody. This is Fernando Ángel Fernández Álvarez from the Spanish Institute of Oceanography in Thom, Asturias. First of all, I want to thank Kat Bolstad for inviting us here. I want to talk about a meeting held in the last Cephalopod International Advisory Council conference. We talk about cryptic biodiversity when we are unable to identify clear differences among different biological species. This can be because actually there are no differences at all among them, but in my experience, this is not the most common scenario. Many differences have been overlooked due to over-conservative taxonomy practices, or because the kind of data that are considered do not cover the actual difference among those species. For example, during the workshop, it was mentioned many times that for some species, it can be quickly identified as different if you look at the alive specimens or just fresh collected ones, but almost impossible after fixation. But at least I can talk about cryptic biodiversity of oceanic cephalopods, which is a topic I have been working for quite a few years already. One of the things that keeps amazing me is how little we know about the biodiversity of animals as charismatic as cephalopods. If you take any oceanic squid family, for instance, the most typical thing is that there is a single species described somewhere in between the last decades of the 19th century and the first decades of the 20th century, which has been cited all around the world. When you test them with molecular methods, most of them usually are more than a single species. Sometimes there are some suspicions, such different morphology on that character no one has been giving it importance, let's say the spermatophore for instance, or maybe biochemistry information. All this information together might challenge or concerns of whether it is a single species or more than one. This is the case of the neon flying squid, for instance. When we look at the molecular information, we discover that there are 4 species within that genus, the genus Omastrephis. Funny thing, I got so obsessed about this squid that I named my kitty cat Omastrephis. Anyway, Omastrephis species, the squid ones, the real ones, are an allopatric species complex, which means that the species distributions do not overlap all of them, making identification based on geography easy. So that was lucky. We believe that the major oceanographic currents maintain Omastrephis populations isolated, so we decided to follow up in this trail, and we test the effect of oceanographic barriers on cryptic biodiversity using 16 squid morphospecies all across the Atlantic Ocean and the Mediterranean Sea, using material collected in several oceanographic cruises. Well, reality did not always align with whatever you are expected to find, and most oceanographic features had little or no effect over the species assemblage we found. However, we did find a strong signal of cryptic biodiversity in 4 of the studied species. One of them, Helicograntia nabose, was in what we call the gray speciation zone. This happens when it is difficult to decide if the observed pattern is the result of high population structure, that means conspecifics, or these populations are in the initial stages of an speciation event. However, there are these few times when a cosmopolitan species actually is a cosmopolitan species. And there are two cases I'm going to comment. Also, they are very charismatic species, so maybe you already know about them. One of them is the giant squid. Up to 20 names has been proposed for that species. However, in 2013, an international team led by Inger Wilkelman discovered that they are a single species, Architeutis dogs. And in a recent and somehow polemic article, the vampire, Vampiroteutis infernalis, was split in two different species. For one of them, a new name has been proposed, that is Vampiroteutis pseudoinfernalis. These two species seem to have a cosmopolitan distribution range. Long story short, is there any way to tell apart cosmopolitan species from cryptic species complexes? Well, you never know until you sequence. So please, feed us with cephalopod DNA sequences. And now I would like to introduce Thom Arnold to you. Thom did a short internship in the lab of Roger Villanueva in the Marine Science Institute of Barcelona when I was a postdoc there. She led research on Ancystrocheirus diversity at the global scale, and she found something so amazing.
Hello everybody, I am Thom Arnold. I'm a marine biologist and I focus mainly on invertebrates and taxonomy. And I will tell you a little bit about the work I did together with Fernando. Our goal was to confirm if there were any morphological differences in the suspected cryptic species complex of Ancystrocheirus lazeri. There was molecular data pointing to a possible complex. Also, there had been a lot of confusion about this name in general. This was mainly due to scientists all around the world describing similar-looking squid in their own way, all focusing on different characters. What all of these specimens across the world had in common was that they had hooks in their arm instead of suckers. They had a purplish mantle and light organs in the skin forming a distinct pattern. To see if these specimens from around the world were different morphologically, we wanted to put them all besides each other. To this end, we inspected specimens from several European museums and collections, using specimens collected in the Atlantic, the Indian Ocean, and even specimens from the Pacific. We also reviewed the sequences stored in two databases, NCBI and BOLD. Based on this data, we found there are at least 6 species within the Ancystrocheirus genus, rather than just one single cosmopolitan species. We were able to resurrect the name Ancystrocheirus alessandrini for one of them, which is present in the North Atlantic and the Mediterranean Sea, and we also characterized the distribution of a few of those species. What is quite unique about this species complex that we're looking at was the arm and tentacle hooks. This was historically used as one of the main classifying features. There is still plenty of research to do with Ancystrocheirus species, but at least we found some morphological characters which are promising to solve this complex. Additionally, within this inspected material, we found a specimen which was really interesting. It had the characteristic arm hooks of this genus, but the hooks looked different, as they have accessory cusps next to the main hook, which kind of resembles a trident. Interestingly, it also lacked many of the other characters we associate with the Ancystrocheirus genus. It lacked any pigment or light organs. Even more importantly, the heart structures, which are really less morphologically flexible, were very distinct and different. After conferring our observations with descriptions of known species, we concluded that this specimen did not only represent a new species, but also a new genus and a new family, which made this the first new squid family described since the 90s. This specimen was found by the cephalopod researcher Malcolm Clark in the stomach of a sperm whale between 1955 and 1956. As this specimen lacked skin pigment and was found in the sperm whale gut, we decided to name them after the Herman Melville's character Moby Dick. And as their arm hooks look like tridents, its specific epithet was dedicated to Poseidon. And that's how the name Moby Dickia Poseidoni was born. Without the continued efforts of natural history museums, this discovery couldn't have been made, especially considering many deep-sea animals have been previously collected with outdated unethical practices such as whaling, which means that the collections we have now will be the biggest source of deep-sea animal taxonomical information we will have for the foreseeable future. This is one of the many reasons it's so important to protect collections. So we would like to end on the note that historical collections and natural collections are so important to protect and maintain because these historical specimens contain so much information and we can still learn so much from them.
Participants in this workshop included people who have or are developing expertise within specific cephalopod groups and their taxonomy and classification. So we've got a couple of these projects going in our lab at the moment. We've got 2 PhD students working on specific deep sea squid groups. I'd like to introduce PhD candidate Austin Horenkamp from the AUT Lab for Cephalopod Ecology and Systematics, or the Squid Squad.
Hi, everyone. My name is Austin Horenkamp, and I am a PhD student here at the Auckland University of Technology, working under Dr. Kat Bolstad. It is a pleasure to introduce you to the family of squids that I work with, known as the Brachioteuthidae. The Brachioteuthidae consists of 2 genera, Sarcicovia, try saying that 5 times fast, and the Brachiotuthis. There are about 7 species between the 2 genera, and it was first founded in 1881. Between 1881 and 2025, not a lot of work has truly been done on this family, so that's where I come in. I am currently doing a global revision of the family using integrative taxonomy. So I am first looking at morphology from museum collection specimens or specimens that are just opportunistically collected if possible. And then I'm comparing the morphology between them. And then I get to take the genetics. I get to do the cool sciencey side and actually look at what genetic differences separate those species.
We also have PhD candidate Ben Shirey, who is working on the jeweled squid family, and he's doing both taxonomic work in that group and some studies on their ecology worldwide.
Hi, my name is Benjamin Shirey, and I'm a PhD candidate at the Auckland University of Technology in the Alice's lab with Kat and Heather. I'm originally from the United States, and I've always been passionate about the deep sea and marine invertebrates in particular. My thesis is looking at the systematics and ecology of the deep sea squid family Histioteuthidae. You may know them a little better as the strawberry, jeweled, or even cockeyed squid. In a nutshell, what I'm looking at is essentially who's in this family and how they fit into the global oceanic food web. With regards to the systematics, I'm looking at several species complexes where we know there is cryptic biodiversity, that being similar-looking animals that have been previously treated as a single species, but genetic differences have now been recognized. To sort this out, I'm using integrative taxonomy, which incorporates genetic and morphological information to help separate these species. I'm describing new species and resurrecting old names where applicable. Presently, I've found some very cool differences in mantle photophore patterns that seem to be extremely helpful in differentiating between them. As for the ecology, I'm trying to get a good grasp on the predators, prey, and overall trophic position of the family globally. As of now, I've compiled an updated list of predators from various sources, primarily stomachs, including important megafauna like toothed whales, various deep-sea fish, and sharks, and more recently we found even other squid. But we still don't know what they themselves eat. So I will be using DNA metabarcoding on stomachs to identify their prey, as well as doing stable isotope analysis on beaks to figure out where they lie trophically. Thank you so much, and feel free to reach out to me if you're interested in strawberry squid or have any questions. I will take literally any excuse to talk about them. Thank you so much.
There were lots of different ecological topics covered. Cephalopods, of course, are very important in marine ecosystems as both predators and prey. A lot more is known about their role as prey because of humans' interest in their predators, such as whales and dolphins and seabirds, than we tend to know about their diets themselves. So a few people are working on dietary studies for various groups. Their diets are very challenging to study for a number of reasons, part of which is they chew their food really finely because, of course, their esophagus passes through the middle of their brain. So it's pretty high stakes if they don't do that, which leaves the stomach contents as a nice slurry of very tiny pieces. And if we're lucky, we might get some recognizable fish otoliths, we might get some crustacean parts, but I can't tell you how jealous I am when you open up a fish and the most beautiful squid specimen I've ever seen falls out of it and could be a type specimen. There's no challenge in identifying that. And we get this smoothie of parts, which increasingly, we're doing integrative taxonomy on these. So barcoding them for CO1 and also morphological IDs. And we find actually that those two methods are very complementary. They pick up different prey items. So with morphology, you get the otoliths and some recognizable pieces. And often we don't pick up those fish sequences when we're barcoding, whereas the barcoding does a lot better job of recovering invertebrates and also picks up some fishes that are probably too large for the squid to consume the head, so we don't get the otoliths. So actually, if we did just one of those methods, we get very different pictures of the diet. And then both of those are very short-term looks at what the squid happened to have been eating right before it was collected. So we have a number of people who are also adding in the stable isotope component for a longer-term picture of what kind of trophic level they're feeding at. One of those people is Christina Fleetwood, who is a master's student at Scripps Institute of Oceanography at the University of California, San Diego.
My name is Christina. I'm a second-year master's student here at Scripps Institution of Oceanography in La Jolla, California. I study trophic ecology of pelagic cephalopods, and I also work here at the Pelagic Invertebrate Collections.
Christina, thanks for having a chat with me. We've actually been in touch for a few years because, as I recall, we met in front of a cephalopod tank at Mbari when you were working there, and I happened to be visiting one day. Am I remembering that right?
Yes, in front of a GPO tank, in fact.
GPO is my first love. That's how I got hooked on cephalopods in the first place. A very fortuitous meeting, as it turned out. And now you're working your way up through the ranks of grad school and cephalopod research. And now you're doing a dietary study on pelagic and some deep sea cephalopods. As with a lot of deep sea stuff, we have to get creative with some different methods.
Yes. So I'm looking at accretionary structures, so their beaks, for instance, and since they're really, really small beaks, I'm having to use the whole structure rather than parsing it out over time. And I'm looking at 15 nitrogen and 13 carbon. So for those of you maybe less familiar with those isotopes, the heavier the nitrogen isotopes, the higher up they are in the food chain. And the 13 carbon kind of tells us where that baseline of productivity is coming from, whether that's more benthic or pelagic, for instance.
It's so cool, right? By studying these two isotopes, you can find out kind of what trophic level they're at. Although there are some complications, right? When we get animals that are scavenging, they can have an artificially high nitrogen signature. And then the carbon one gives you a bit of a read on location. So you get this really nice paired data about how high up the food web they're eating and where they've been. And for larger beaks, you can do this more across the lifespan as well. So you can get a read of sort of a history of what the animal's been doing. And so you're looking at the beaks of really tiny ones, and beaks are tricky to get out anyway. So I have a lot of respect for you doing this with really tiny animals. Which species are you looking at? What are you finding the most?
For the first part of our study, which was conducted across the Santa Lucia Escarpment, we got primarily chirotuthids, mostly paralarvae, as well as histiotuthids or strawberry squids and vampire squids, as well as lots of other taxa, but those were more 1 and 2 offs.
Oh, now vampire squid, that's very interesting. Off the top of my head, feel like I read something that suggested that vampire squid, which are at least partial detritivores later in life, maybe start out as more active carnivores and become detritivores? Am I remembering that right?
That's what I've heard anecdotally, so I haven't opened up their guts because they are rather small. I will say, just having a preliminary look at the data, they are showing up very high in the food chain. Everything dies, so everything will be part of marine snow, and that might kind of artificially inflate their trophic position.
Now, when you say small ones, have you seen any of the awkward teenage stage vamps that have 4 fins?
Ooh, no, I don't think so, but I will say that they didn't come up in the best shape. So even if they did have 4 fins, they might not have had 4 fins by the time we got to them.
Okay, we'll have to post a link to this. There is an absolutely adorable clip of a vampire squid. And for those who don't know, vampire squid babies have a pair of fins that they partly use to move around with. But as they mature, those fins regress and they grow a new pair of fins that has a different gait that it provides to the animal. So they use these 2 pairs of fins quite differently. And for a very brief, adorable, sort of dorky, awkward teenage phase, they have 4 of them, and they kind of work at odds with each other. And so there's this little video clip of a baby vamp in a tank that's got its 2 pairs of fins going, and it is honestly one of the cutest things I've ever seen. How far through your study are you now?
So I kind of have 2 parts to my research. So like I mentioned, the Santa Lucia Escarpment, I was fortunate enough to go out to sea this past February, March, and take some trawl samples, and the rest of our crew was taking zooplankton and physical oceanography samples, just trying to characterize this region. That part of my study is a little bit further along. And then I was also fortunate enough to get bycatch from a NOAA survey that just came back a couple months ago, the Coastal Pelagic Survey. So they survey the entire west coast of the US, all the way from San Diego to the top of Washington state. So I'm still kind of processing and pulling out the beaks from those animals as well.
That's kind of a common theme, right? Cephalopods, especially not the non-commercial ones, are really difficult to target with research gear. So a lot of the samples we get are just incidental bycatch from people who are studying or fishing for other things. And that's one of the reasons we tend to have fairly low sample sizes, is that, we just get the odd unlucky one that ends up in the net with everybody else. But it's always really cool then to make use of those, right? So that's not a complete waste that animal happened to get caught. We at least can use that to learn something about it. Any initial findings from what you've been doing that you'd like to tell us about?
Well, so far, no big surprises. I'm still in the very preliminary phase of looking through data, but we did see kind of a big grouping of all of the cephalopods across the escarpment, with the exception of a couple muscular guys and then the vampire squids, of course. Everyone else is pretty much the same trophic position or within 1 or 2 trophic positions of each other. So we're going to explore whether or not different species are migrators, where they exist in the water column. And these trawls were going on throughout day and night. So there is a little bit of parsing out that needs to be done with respect to time. So it'll be interesting to see how these animals kind of living on top of each other are all feeding very similarly.
And do you have much of a size range, or do you have some that are larger or older than others and sort of overlapping cohorts?
I would say there's a couple that I would be comfortable saying are adults, an Octoboteuthis and a Gonatopsis, but everyone else is pretty much a paralarvae or subadults. So they'll fit in your hand. They're pretty small, and then a few are very, very new paralarvae.
Of course, as they go through those different life phases, a lot of these animals change their diet along the way, not just size classes, but completely different taxa. So really interested to hear, because some of these ones that you're studying, this will be the first dietary information for them ever. So anything that you can contribute there will be really valuable in helping us understand the biology of these hard to get to species.
Definitely. And also kind of funny that they're in such a well-studied system, right? The California curry system is a highly researched area, and still there's a lot we don't know.
Yes. Relatively speaking, when we say highly researched area of pelagic or deep sea, that is very different from a highly researched stream or highly researched forest system, right? It's,
of course.
Any takeaways from kayak that you found really cool?
Just being around cephalopod people is really fun. Here at Scripps, where there are only 2 of us working on squids in general, and we do very, very different things. So kind of being around a community that understands what you're going through and can talk through the nitty-gritty is really, really helpful. So I'm in a zooplankton lab, and it's very helpful for me kind of looking at the overall trophic ecology of the system. But I don't have as many of those squid specific conversations. So that was helpful to get a bit more perspective about what people are working on and how it kind of lines up with other parts of the world.
It seemed like a lot of the early career researchers who were there, the grad students and the recent graduates, there's a really nice community in that kind of career stage. And it just seemed like, people were having a great time making connections and building those relationships with their future colleagues, which is great to see.
Definitely. I think it helps a little bit with imposter syndrome to kind of hear that people are struggling with similar things. We're all kind of at the same place doing different things, but that was, I think, very useful to experience.
Imposter syndrome, fortunately or unfortunately, doesn't necessarily go away. Those of us who have been doing it for a while, we're maybe just better at hiding it. It doesn't mean it's not there.
That's fair.
Thank you so much, Christina. That was great.
Thank you so much for having me, Kat.
We also have someone in our lab, the Squid Squad, who is studying squid diets locally using this same combination of factors: morphological IDs, CO1 barcoding, and longer-term stable isotope. With those, he's investigating the diets of 5 deep-sea squids whose diets are either completely unknown or have not been investigated in New Zealand before. So this is Sam Clough from the AUT Squid Squad.
Part of my PhD research focuses on the dietary niches of the Angolan and the Antarctic flying squids. These sister species both look quite similar. They're about 1 meter long and weigh about 3 to 4 kg. These 2 species are so similar to each other, we would expect them to exclude one another, yet both co-occur in New Zealand waters. I hypothesize that the mechanism behind their coexistence is that when these two species co-occur, they select different types of prey to avoid competition with each other. To investigate this hypothesis, I've been identifying prey species from the stomach contents of the two squid species. Yet this method only provides a snapshot of what they've recently eaten. To overcome this bias, I have been analyzing the nitrogen-15 and carbon-13 values of the squid's beaks. To put simply, the different prey groups have their own nitrogen-15 and carbon-13 values, which are absorbed into the squid's beaks when it eats them, making the squid beaks more like the nitrogen-15 and carbon-13 values of the prey it regularly consumes. So what did I find? Both squids had a lot of lanternfish in their stomach contents, which are these tiny fish that rise from the deep sea to feed on planktonic crustaceans at the surface at nighttime. Both species also had their fair share of the larger deep-sea fish, such as hoki and rat-tail fish. Despite the overlap, the Angolan flying squid had a greater diversity of small cephalopod species, such as the gelatinous telescope octopus. Based on stomach content analysis, it would seem that their diets are quite similar to each other. However, nitrogen-15 and carbon-13 values tell us a very different story. The Angolan flying squid had values more like the small squid and lanternfish we found in its stomach, whereas the Antarctic flying squid had carbon and nitrogen-15 values more comparable to the larger deep-sea fish we regularly found in its diet. So it seems that despite the stomach content analysis overlap, the two flying squid species do diverge more than stomach content analysis would initially indicate. Thanks for listening to me talk a little bit about my research. If you want to learn more about my research, you can find me on ResearchGate, and also check out Fernando's online talk if you'd like to learn more about the biology of flying squids.
If we want to give people a hand with sort of trophic levels and stable isotopes, a nice little primer on that is there are different isotopes of some common molecules. There are some that are a little bit heavier, and biological systems seem to like those heavier ones. They integrate them into things at a slightly higher rate than they're found in nature. And so every step of the food web, you end up with a little bit more of these ones. So the ratio changes. It's really interesting that we can use such chemistry to find out, well, how high are you in the food chain? Who's eating who? So that's where the sort of trophic and the stable isotope side of things come. And now that we've got genetic barcoding, again, we can sequence the goo and find out who is in the goo. So those two approaches are letting us know where they sit within the food web.
Yeah, and there were some really cool talks about different types of chemistry. So the beaks of squids and octopuses are one of the structures that people tend to use most often for these stable isotope analyses. There are trace elements recorded in the statolith, which is the squid version of the otolith as well, and those have really well-defined growth increments. So if you know how often those growth increments are laid down, you can actually get a really detailed read on the squid's life history. And as you mentioned, those nitrogen isotopes give us trophic information. You can also study carbon isotopes, which give more of a geographic or depth profile. And then if you have lots of funding, you can study oxygen isotopes, which give you an indication of where in the water column the animal's been. And that gives us some interesting insight into maybe ontogenetic descent from those early life stages in the shallow waters down into the deep sea later on. And we had someone speak about the fact that you can actually recover quite a lot of hormone information from beaks as well. So they're starting to get some further insight into physiology and life cycles based on studying those from beaks. People are using historical collections from museums to track past climate fluctuations as well. Few people are looking at the chemistry of the embryo and its surrounding fluid within the egg and looking at whether the depletion in oxygen across development is what eventually triggers hatching. The oxygen levels just get so low inside the egg that the squid has to hatch, and then it actually gives a visible little gasp as it comes out. So lots of really cool ecology and chemistry stuff. We have another interesting aspect of ecology running in the squid squad. So student Marina Maranzana from our lab is just about to submit her thesis about symbiosis, so about the microbiota associated with deep sea squids. I know you just spoke with Margaret McFall-Nye. That was an awesome interview. Marina is studying a far less well understood system of who lives in association with the deep sea squids.
Ciao, I'm Marina, a PhD student at AUT's Lab for Cephalopod Ecology and Systematics, also known as ALSES. I study the microbiota of deep sea squids. This means I look at the different bacteria that live on and inside these squids. For my PhD, I focused on 9 different species of squids, all within the order Egopsida, including the colossal squid. The bacteria that I have found is quite different than the bacteria that has currently been reported in coastal squids. This is especially true for bacteria found in their reproductive organs. We think this might be because of the different egg-laying strategies between deep sea and coastal squids. Coastal squids are known for laying egg masses on the sea floor, and from what we know of deep sea squids, they tend to brood their eggs in their arms or lay floating egg masses. Therefore, they might need different bacteria to protect their eggs. Given these initial findings, it would be interesting to look at what the functions of these bacteria are. Although the reproductive organs of deep sea and coastal squids harbor different bacteria, the different genera of bacteria might be performing similar functions. Thank you for having me, and keep an eye out for any future publications if you're interested.
So all of these ecological studies are interesting because they're providing insights into these animals, sometimes, for the first time, even for a species within the whole family. It might be the first time we know anything about that family. But as we know from many different deep sea groups, what we're finding out now may or may not represent a baseline of how things were before humans started to mess with stuff, right? Because we're learning this stuff for the first time, but the oceans are already changing. So we had lots of talks about the effects of temperature and acidification on different aspects of cephalopod biology. There was the sort of perception that cephalopod stocks were expanding worldwide because the overharvesting of their competitors and predators, specifically large finfishes, has left some vacancies that cephalopods can rapidly fill. But it turns out, that some of these rapidly warming and acidifying conditions are not great for them either. So probably did have increasing populations in some cases for the last couple of decades, but some of those are starting to come back down again. But we're also seeing some changes in range, like famously the Humboldt squid's range has expanded. And there was a very interesting talk from Mike Navarro about new range expansion of a deep sea squid into the waters outside his laboratory.
Right now, I'm speaking with Dr. Michael Navarro, who's an associate professor of marine fisheries at the Department of Natural Sciences at the University of Alaska Southeast. Thanks for joining me, Mike.
Hi, it's great to be doing this podcast. Thank you.
You talked about a really cool thing, right? So in the era of some of the changes we're seeing in the ocean, one of the big questions is how our species range is changing. And you told us that you're seeing some interesting changes in your local marine ecosystems that involve deep sea squids. So can you tell us what's going on?
Sure. Here in Alaska, as far as for those squid that have the potential to be fishery species, that includes Paroteuthis opalescens as well as Paroteuthis magister. One of those is endemic. That's Paroteuthis magister, or the armhook squid, or magister squid, as they're sometimes referred to. And their abundance seems to be increasing a bit. That's at least what anecdotally our fishermen have been talking about over the last few years.
Yeah, and these are squids from the family Gonadidae, right? Which is one of the typical deep sea squid families. Like some species are known to carry their egg masses and brood them. As a person who likes to keep track of egopsid or oceanic squid sightings on iNaturalist, I definitely have seen, I think, an increase in the Paroteuthis sightings on iNaturalist as well. So this ties in with some of the local fisheries that you have going on. How are the local community responding to these changes, and what might happen with the fisheries as a result?
Well, our communities are really curious about the Paroteuthis magister. They have been seeing them in their catches. We have, as you can imagine, Alaska has, our communities are very interested in all kinds of fisheries. So we have sablefish, salmon, of course, halibut. And for sablefish and halibut, these can be deeper sea fisheries. So people can go down to 100 meters or more sometimes to fish for them. And when they do, every now and then they'll pick up some squid. And that's where I think a lot of the curiosity and the interest is, because people want to know, what is it? Why is it here?
But there hasn't been a fishery for this species in the past, right? Not an appreciable one.
No, that's exactly right. No commercial fisheries. There has been, in general, you can catch squid. But it's not even species specific. That's the particular policy. So there is interest in our community now from fishermen in developing a commercial fishery for Paroteuthis magister.
You mentioned some really interesting sustainability requirements that all fisheries locally have to meet. Can you tell us a little bit about that?
Yes. In Alaska, it's in our state constitution, it's actually, we are required by law, state law, to have a sustainable yield principle, which means that in order to harvest anything, there needs to be demonstration that it can be done sustainably or to the sustainable yield principle. That means when somebody proposes a new fishery or any kind of resource extraction, natural resource extraction, then demonstration that it can be done so sustainably.
Right. So as a prerequisite for getting a new fishery established, you have to show that it will be sustainable from the beginning.
Correct. Yep. And then early in 2025, there was a proposal that was put in place for Beartooth's Magister. And to kind of give an example of this, that proposal was not accepted because of a lack of biological information.
Mike, somebody should study this squid.
So that's what really, with encouragement from the community, working with the state, we're doing a little bit of that here at UAS Southeast, and in particular, looking at these harvested squid for a number of biological characteristics, basic characteristics such as size, reproductive maturity, etc., as well as aging, and then kind of getting a little bit into what they're eating and those kind of.
This family of squids, the gonadid squids, there are quite a few species that occur in the North Pacific, and I know they are fished in some places. One of the convenience stores I was in Japan for this conference, there was a packet of dried squid, and looking at the photo on it, I could tell that it was a gonadid squid because it had the right unique armature on the arms that were visible in the photo. So that was a first for me, being able to ID something based on the photo on the package for a deep sea squid. Maybe that means that, there'll be some published fisheries information for related species that you could draw on as well.
Yes. In fact, Japan is one of the leading, their teams there, as well as Russian research teams are some of the key scientists for this particular species. Our B. magis has a range that extends from Japan all the way through the North Pacific down into the States, through Canada, etc.
Nice. Yeah, I mean, it's certainly not one that we're familiar with. We only have one species in that family down around New Zealand, and that is one that is mostly Antarctic, but does occur a little bit further north sometimes, but it's a real distinctive one. In my pages and pages of notes I took at Kayak, I was looking over cool stuff you had said in your talk. And I think you were the person who mentioned that the embryos of cephalopods that have large eggs can see prey while they're still in the egg, and that's when they develop their search image. Did you say that?
Yeah, that's correct. Yeah, they basically are born into this world ready to hunt.
That is amazing that they can see through the egg capsule and well enough to start knowing already what they're looking for when they hatch and start hunting.
I tell you what, when I go fishing, if I don't catch anything, I still get to go home and eat. But like for these little baby squid, they have to be able to not only hunt, but successfully hunt within a few days, or else they run out of yolk.
That's amazing. And there are both some squid and some octopuses that have relatively large eggs. And picturing those embryos, that makes sense to me. Now, of course, I'm wondering for the real deep sea ones that have those large eggs, if those babies are also still hunting visually, and if they have this development, because, like Megaloletony, the big Antarctic deep sea octopus, those eggs can be like 4 centimeters long. Those are pretty well developed babies when they hatch. Now I wonder what they're capable of as soon as they come out.
Yeah, that is interesting. That is big. That's
huge eggs. I couldn't believe we had a female specimen and some of the eggs came out. I was just like, those have got to be the biggest cephalopod eggs, by orders of magnitude. It was amazing. I've really enjoyed your talk at Kayak, and I'm glad to be able to follow up with you on some other projects.
Wonderful talking.
So in this changing world, cephalopods are rapid adapters to environmental change, but some of their biology is particularly interesting because they have the ability to do some pretty neat tricks to assist with these rapid adaptations. So I know octopus RNA editing has been an interesting topic for a couple of years.
Because they're aliens, right? Because of the aliens thing? Because how they're aliens?
Oh, you know what? I know somebody we should ask about whether they're aliens. Let's ask Meg Midland.
Hi, my name is Meg Minlin. I am a molecular biologist and science communicator, and I study octopuses and a molecular mechanism that they do called A to I RNA editing. And my socials are Invertebabe. The only different one is Instagram, which has a dot in between in and Vertebabe.
So thank you so much, Meg, for having a chat with me about your research. You're also one of the main organizers of the Ceph Research Network Discord, correct?
Yes, I am, though Austin started it, and so I think he deserves most of the credit.
You're doing a project on octopus RNA editing, and this is really a cool topic. I learned a lot about it at Kayak, and I'm excited to have you tell us a little bit more. It is one of the things that people who do SciComm have to myth bust quite a lot. So I'm going to come right out with the question you probably hate, which is, Meg, Are octopuses aliens?
It would be really fun if they were, but they're not. And it is a thing I have to talk about a lot because my research and other scientists who do RNA editing research have kind of been put in the crossfire for this question because there is an academic paper out there that puts up this hypothesis that octopuses have come from a meteorite from outer space and uses RNA editing as the evidence for this claim. It's a very bad paper. It has not been peer reviewed, but nonetheless, it's spread like wildfire all over the internet. And, with some understanding that, people know a lot about octopuses and they're really weird and they're considered the only intelligent invertebrate. So it's not like far off on people's radars to really believe this. But they are not aliens. I think the thing that annoys me the most about the claim that octopuses are aliens is that I think it is so much cooler to be as weird as they are and to be made of all the same materials that we are made of, which is part of the reason why I love RNA editing. It's because it's a molecular mechanism that everybody else uses, but it is utilized in a completely different way in cephalopods and a way that we had not imagined before. Because I love sci-fi, I love fiction, I love the creativity that people put towards the ocean and deep sea animals and what fuels a lot of our narrative surrounding, deep sea animals being aliens. A lot of our creatures are inspired by them. But nobody ever thought of RNA editing. No one ever thought of, wouldn't it be so wacky and interesting if some animal edited their RNA to change their proteins, to change the way that their bodies communicate to each other as a tool of genetic editing instead of just changing your DNA, which every writer out there has written a story about. And so I love that for them because it's just so interesting and unique and something that humans had not yet thought of until we found it.
Such a recurring thing in deep sea, right? Is we come up with all of these wild sci-fi and fantasy creatures, and then it turns out there's all this stuff in the deep sea that goes even way further beyond sort of the wildest imaginings of what we've come up with. They're real, they actually exist, and it's even more bonkers than what we've invented.
No, it's part of the reason why I love the deep sea because so much of our stories come from the deep sea, from artists being inspired by things in the ocean and then writing really funky stories to go along with it. And I think the deep sea just fuels people's creativity and imagination. And I'm really just kind of sad that imagination spread into disinformation on the internet instead of I don't know, a cool sci-fi novel. Once we get down to the really weird, kooky things that animals do, it's just oh my God, I can't believe that this is a real thing.
Yeah. This shares my planet, and we share ancestors with this thing.
That's why I think RNA editing is so cool because, man, they did something completely different than us with the same materials.
For those of us who have vaguely heard about RNA editing as a concept previously, but don't have a deep understanding of it, can you tell us a little bit about generally what it is and the fact that lots of animals do it, but then what it is that octopuses are doing that's different?
The way I like to preface my RNA editing explanation is that we kind of all know animals evolved through random mutation to their DNA. And that's kind of how we acquire traits and change things about our bodies, and in very simple terms, become newer species and organisms. There's a second step to DNA that people often forget about, not anymore because of COVID, RNA, which is our intermediate step to getting our proteins, which is all of the things that work and make your body work. And the way I like to think about it is that DNA is going to be the cookbook to make all the things that work in your body. And RNA is going to be all of those little individual recipes. Sometimes, generally, a mutation arises in your DNA, and you can think of that as swapping out recipes or, maybe changing the cookbook altogether. But what octopuses and squid and cuttlefish are doing is they're tinkering with the individual recipes themselves, the RNA. And what that ends up making is very similar versions of proteins that act, slightly different from each other. So it's like if you have a chocolate chip cookie recipe and you change out the amount of flour or the amount of salt, tinkering with those little individual ingredients, you still get a chocolate chip cookie, but you get a chocolate chip cookie in a bunch of different textures. Like you get a cakey one, you get a chewy one. There's just lots of different ways to make a chocolate chip cookie. The way that octopus, squid, and cuttlefish are doing this is they're tinkering with the little individual recipes and slightly altering the proteins in their body as a result. And so the way we've seen this is by studying temperature and how warmer and colder water affects editing, because it's happening all the time, but it's easiest to change a variable, so like their environment, and see what's edited differently when you're studying something like this. And what they found is, it's things like the rate of ions moving in and out of a channel, the rate of kinesin, which is a little motor protein walking across a little microtubule, and even like conformational changes to proteins like synaptogamma, which is a nervous system protein. And so all of these changes are happening in their brain, and it happens in the rest of their body, but just more so in their brain. And the commonality is that they seem to be changing the rate of their brain cells communicating to each other. So just making little changes depending on the temperature that either speeds up that communication or slows down that communication.
I know that you mentioned a little bit about this with implications for climate change. I'm, of course, thinking about, whether and how much we know about this for deep sea stuff. So I know you study animals that live in fairly cold water in the Pacific Northwest. Do we know anything about these mechanisms in the deeper sea species?
We don't, which drives me wild because I want to know. The thinking is that these changes that cephalopods are making to their proteins can enable them to better acclimate to their environment. And so if you have an ocean that's constantly changing, which is, an ocean that they've lived through, then it's a really great thing to be able to edit their proteins to acclimate to those changes.
If you have a wider range of ingredients or just variations available, the more likely you are to have something in your toolkit that lets you survive a change, right?
Yeah, exactly. And so I'm studying this in regards to ocean acidification, and we're seeing how a transcriptional factor, zinc finger proteins, which repress or activate genes.
Of course, that's exactly what I thought they did. Yep.
Yeah, so that's how they could. A little bit too advanced in my terminology, but that's how they can. I've linked my research to behavioral changes that cephalopods make in result to increase acidification. And so the idea is maybe that this is helping them out a little bit. But why this is so interesting with the deep sea is that in the deep sea, you don't have as much of a constantly changing ocean. The environmental conditions are a little bit more stable. They're not like the warmer oceans that have gone through warming and colding and increased acidification and lower acidification. There's a little bit more stability. And so my question has always been well, is editing still happening then? Are they still needing it?
Yeah, because I always make this point as well, right? The deep sea is an extreme environment in a lot of senses. In other senses, if you compare that to a tide pool, a very different type of extreme. It is an extreme set of circumstances, but with very, very slow rates of change in most places. Not the same as it changes on a 6-hour tidal cycle and goes from completely dry to flooded and heavy wave action. So yeah, so maybe under these more stable conditions, these rapid adaptive mechanisms are not as needed, and we don't really know whether they persist or not in those taxa.
Yeah, but then also the interesting thing is we know that the original RNA editing paper was done with an Arctic cephalopod. That's a very cold water, I think kind of deep living octopus that they do found RNA editing rates with. And then also editing is suppressed in both warmer water and more acidic water. Okay. And so the deep sea is, right, it's more acidic, but it's a lot colder. Editing could still be happening, but then what's going on? What are they editing if that is happening?
Yeah.
I don't know. There's just a lot of questions.
One more question of what are they doing down there, right? We have so many questions about this. Interesting to hear you refer to the original paper that caused all of this trouble with octopuses being aliens. I am shocked and dismayed to discover, maybe I'd forgotten that was an actual paper that someone put out, because I always thought it was someone had said they are the closest thing to aliens we have on Earth, and that like the media had just mangled that into that sound.
They did
octopuses are aliens. I know that happened as well.
Oh yeah, it was published by a bunch of astrobiologists, and there was no zoologist on the paper. There was like a huge amount of people on the paper, but no zoologists. And you could really tell once you got into the octopus section, because they are also like, oh, well, octopuses appeared out of nowhere and we have no idea their evolutionary history. Oh man, like the way you just ignore an entire...
They don't fossilize like trilobites, but we know where they came from.
Yeah, but we do know where they came from. And then it was also like they posed a hypothesis that squid turned into octopuses from a virus that came from outer space, which is a very funny graphic that they put in. It looked like it was made like a 10-year-old. It's a very funny paper, but also a very, oh my God, I can't believe this got published in an academic journal and that it is going to haunt me and other octopus scientists for the rest of our lives.
But, one of the things that this podcast is really good for is myth busting about deep sea stuff and associated things. So, well, thank you so much, Meg, for having a chat with me. You do such cool work, and I can't wait to hear when we get more insight into what's happening in the deep sea. Maybe we can work on that together. And thank you so much.
Yes, thank you so much for having me on this podcast. I love it.
I'm disappointed. I wanted them to be aliens.
Well, I think a lot of people did, and that's why it got the traction.
Yeah, that's how it is. But the pyramids were made by aliens, so it's all good. Discovery Channel after 8 p.m. It's all Nazis and aliens. Sometimes both.
What a party. As cephalopod populations are expanding, contracting, changing their ranges. Oh, someone introduced the term teuthogeography, which I really liked. So that's using cephalopod biogeography to infer other bits of information about oceans and food webs and trophic
really
systems.
Mobile, rapid generation time. Historically, things like that have been done on like the brittle stars and things like that. But that's not going to respond rapidly to a changing world, whereas these are quite mobile animals.
Yeah, and there was a really amazing keynote actually by Professor Jan Strugnell from Australia at James Cook University, who used the distribution of current deep sea octopus species around the Western Peninsula and Antarctica to infer when the ice shelf last completely collapsed, some differences in sea level at that time, and to also predict what might happen under future warming scenarios. So we'll link that paper in the show notes. As cephalopod populations and distributions change, of course, humans continue to harvest them, right? They're an important protein source in a lot of parts of the world. They are quite a challenging group to manage sustainably because their populations change so rapidly that it's almost impossible to get a good estimate of biomass or abundance before you start fishing in any given season. So that leads to some challenges in managing them worldwide. And in some places, multiple overlapping species are fished and managed as a single fishery. So to tell us a little bit more about that, we've got PhD student Connor Wallace from the AUT Squid Squad.
Kia ora koutou, and hello everyone. My name is Connor, and I'm also a doctoral candidate of AUT's awesome Aussies lab. One of the things that I'm most passionate about is conservation, and so I've come into cephalopod research with a bit of a different background from some of my peers. I completed a Master of Science project looking at viruses in New Zealand native parrots. Our kakariki and kakapo have done research identifying terrestrial invertebrates in a mammalian predator-proof sanctuary and have worked to protect and monitor Peka tauroa, Aotearoa's endemic long-tailed bat. I've always found cephalopods fascinating and was looking for a PhD project that would allow me to combine that fascination with my conservation and species management background. Side note, if you're pursuing biology ecology research, a lot of skills are transferable between different study organisms. So don't be afraid to change things up a bit and pursue the research that inspires you the most. Nowadays, I'm researching Aotearoa's arrowsquids, two omistrophids in the genus Notododurus. If you can picture a squid in your mind, these squids are very squid-shaped squids with a torpedo-shaped body and arrowhead-shaped fins. The aim of my research is to improve our understanding of population structure to improve the sustainability of New Zealand's arrow squid fishery. They're significant species for people. In New Zealand, we catch about 30,500 tonnes per annum, which is worth roughly 140 million New Zealand dollars per year. And they're also ecologically significant as prey items for animals such as the longfin pilot whales and the endangered pakake. or New Zealand sea lion. When we're looking at conservation and species management, one of our goals is to build the best picture we can of the life history of the species that we're interested in, the survival pressures it's under, and to understand how populations of the species are structured. With fisheries, population structure is super important. If you're fishing in one spot, thinking that the squids in this area are only a small part of a larger population, and then it turns out that there isn't a larger population, but actually two completely separate ones, you could overharvest a population, losing genetic diversity and collapsing a localized food web. Aotearoa's arrow squids typically live up to a depth of about 300 meters. And with deep sea animals like this, it can be really difficult for us to understand population structure. We can't directly observe how many squid are present in the environment. We can't attach GPS trackers to log the dispersal of individuals and measure population connectivity. Furthermore, the rapid generational cycles that we see in many squids can make it hard to assess biomass. Fortunately, genetic tools offer a possible way forward, and so this is what I'm pursuing with my research. Genetics techniques can provide clear measures of population relatedness and don't rely on the observation of live specimens. A small tissue clip can be all the biological sample that you need. I'm fortunate to be working with a wealth of arrow squid tissue samples collected by Earth Sciences New Zealand, and I'm using a combination of microsatellites and single nucleotide polymorphisms, two different genetic markers, to look at how related to each other arrow squid are around Aotearoa, New Zealand. Ideally, with this relatedness data, we'll get a picture of where populations stop and start and how they mix.
One of the topics that's been very interesting to see develop across the last few cephalopod conferences is what kinds of technology people are using to study these animals. They don't lend themselves well to being studied in a lab, especially if you're studying deep sea ones. People are making some very interesting behavioral observations in the wild, and then some of those species can be studied in situ by bringing them into the lab, but it remains pretty challenging. You know, people are doing things like for the octopus species that's known to hunt together with a group of fish, someone has now developed a robot fish whose behavior they can control to test how the other members of the co-hunting group respond to different prompts given by this robot fish.
Wow.
That was one of my favorites, actually. The robot grouper is pretty cool looking.
I like the interspecies collaborations.
Mm-hmm. I mean, I know you need a robot fish. Like, as soon as I saw that, I was like, oh, I know somebody who needs that. People are doing some really amazing stuff with technology. We have people who are able to map the development and cell migration within embryos and like track where different parts of the early embryo end up. Watching the cells migrate in real time by coloring them differently and seeing across the first couple of days of development which parts of the early 8-cell stage turn into which parts of the brain and body. And in terms of studying them in situ, people are making advances in how to study these animals in their natural environment as well. There was a cool talk about tagging 3 different species of animals that live within a Spanish MPA and monitoring using a moored array of hydrophones how those different species movements were tracking in real time. So I'm going to pass over to Tony Sanchez Marquez.
My name is Antoni Sanchez. I am a PhD candidate at the Instituto de Ciencias del Mar in Barcelona, Spain, and I'm currently working with deep sea, especially deep sea marine protected areas. And in this case, we're trying to get to understand how different species use the territory or the marine protected areas themselves. So we are doing some kind of tracking methods to evaluate how they use the territory.
You actually have got a setup where you're able to do some experimental work, which is really nice. So can you tell us a little bit about the study area where you've been working and the 3 species you're looking at?
We've been working in an MPA near Barcelona. It ranges in a depth from 325 meters to 550, more or less. The area is characterized by muddy bottoms. It doesn't have any hard structures for the individuals maybe to lay eggs on or for corals to build themselves, because the area has been deeply impacted by trawling activities. So all these hard structures like potentially rocks or other things have been removed from the areas.
And you're interested in 3 particular species that are co-occurring there. So who are they?
We are working with Nephron norvegicus, which is the Norway lobster. This is like the main focus of my group during the last maybe 20 years, and now we are kind of expanding. So this species is an ecological engineering. So they provide a little bit of movement in the sediment. They construct burrows and they give a little bit of tridimensionality to the area. And on top of that, we also wanted to study two of their predators. In this case, we're working with the small spotted cat shark, Silurianus canicula, which is a highly mobile demersal shark in comparison with the nephros, for example. And on the other hand, we're working with a little octopus, Eleganophirosa, or a curled octopus, which is an octopus that covers themselves in sand, doesn't move that much in comparison with the shark, and they also prey on the lobster. And it's quite interesting because we selected this species based on the phylogeny of the species, as we wanted a wide range of coverage. We also wanted to work with Atelos fish like conger or European conger eel. But in this case, we were not able to catch any individuals during the experimental trollings. We also tried to catch them with baited traps, yes, but it was highly time consuming, so we dropped that species.
So those are three really different types of animals, right? And really different considerations for tagging. So you've got a shark, a lobster, and an octopus, and you have managed to figure out how to catch, tag, and then track what they're doing. So can you tell us a little bit about the tagging and tracking methods?
For the tagging methods, we work based on previous literature on the topic. So the most obvious case would be the lobster, as is the main focus of the study. So in this case, we conducted an experiment some years back in a similar area, in this case in the northern part of Catalonia, which would be Palamos. Here we tracked 50 individuals, I think. And the idea is that as they are highly mobile, they use their tails, so we have to tag them on the cephalothorax with a little bit of epoxy to secure the tag, and also a bridle. So it would be like a little backpack, which they carry. And the good part is that when they molt, they leave the tag, so we are not giving this individual an assured death to maybe problems shedding or things like that. They usually shed once per year in the case of the size of the animals we tag, so we are sure that the species or the tagged individuals will continue with the life cycles. In the case of the sharks, we can catch them easily, and we can do a little surgical procedure. So we did a little cut in their stomachs. We introduced the tag, which is like a aspid, would you say, size-wise. We give them 2 suture points. The chance of survival after the procedure is nearly 100%. And finally, in the case of the octopus, this was the trickiest one, because as we know, they don't have a hard patch. And in this case, we based our method on some previous studies that were conducted in Galicia, I think. In this case, they try to tag the individuals in different areas. The thing is that when they clean themselves, they usually grab anything that is not theirs and start pulling.
Yeah, so first we have an animal that's really squishy and soft, and second, an animal that's very dexterous and good at grooming itself. So,
exactly.
This is building through different layers of very challenging tagging technology.
And in this case, the solution was to tag them in the left arm number 3 in the near the base. They wouldn't have any problems using that arm. In this case, putting it in near the base makes it harder for them to grab onto it and try to pull. The lifetime of the tag would be like between 3 and 6 months.
So you've got 3 really different benthic species living in this soft sediment environment, and an array of moored hydrophones, right? And I think you said they have overlapping ranges, so you've got pretty good consistent coverage through your study area. What patterns are you seeing?
In this case, we had some previous studies on nephrops, so this species is highly territorial and also construct burrows. So we expected them at the beginning of the experiment, after the release procedure, for them to try to establish themselves. Some of them will leave, some of them will establish properly, as there are other lobsters already living in the area. So that's one thing. And later we see that these individuals, once they establish, they don't move that much. So they would be within the 3 species, the least mobile, they will be territorial, they would exhibit the most site fidelity of the 3.
Yeah, they stay the most localized.
Exactly. In the case of the octopus, we observed that they move way more than the lobster, unfortunately.
Did they stay within the study area, or did some of them exit the study area?
Some of them did exit, some of them stayed, but as we tagged, I think 14 individuals, most of them left the area. Between 3 and 5 stayed, but they stayed the whole study period, which lasted 6 months. In this case, 4 for them because it's the lifetime of the tagging. And finally, the sharks, they were the most mobile of all. As we cannot encapsulate the whole MPA, we encapsulate a little proportion, like 1/4 of the area. We observe how they go in and out of the array. Although they move a lot, they are also exhibit this kind of side fidelity.
Okay, so they move around a bit, but maybe they have sort of a home territory. It's just a bit larger.
And they probably overlap between individuals. It's not like in the case of the lobster where you have one individual which would be focused on a little area. In this case, the individuals come and go. Yeah, they not exclude the other individuals from their territory. This technology allows you to position your individuals if 3 or more receptors detect the same signal. If we are working usually in shallower areas, like 20, 30 meters, the data quality is very high. So you can track individuals very precisely during long time periods. And you can also do this kind of studies where if 2 individuals get too close to each other, you can like to establish an interaction, but our data has not that quality. So we weren't able to do this kind of procedures.
How far through the study are you? You've finished all of your data collection now, or do you have more seasons coming up?
Yes, we have finished all of the collections. What I presented of kayak was one of the chapters of my thesis, first 6 months of the study. In December 2023, when we collected this data, we also redeployed them and we also redeployed another 70 individuals. In this case, we successfully tagged 3 European conger eels. We also tried tagging some other bony fishes. Unfortunately, didn't get some quality data from them. So we are not sure if it's related to mortality or because they left the area. But yeah, the following study that we do will be working with the whole dataset. So it would be a 1 year period. And we would like to see if there are changes in the different seasons or maybe monthly in how they use the territory, their TL activity patterns, if they shift depending on the light hours or other parameters. The BL activity patterns of the species based on the movement data that we collected doesn't match with was previously known because in this area there's a lot of trolling. They did some studies some years ago which were like short trolls in different hours of the day, so we know when some species are more available than others. We would like to see how this changes during this whole year period. In this case, these trolling experiments were done within a month. So maybe in this case, they touch just a little variability within the whole year.
Yeah, so within a short time period within a particular season, and they only captured that particular seasonality.
Exactly. So we have to look into that thing and would give us a lot of information about the fisheries and the species of the area.
It's always interesting when you go and you're able to collect a larger data set and that contradicts some little snapshot that was the basis for all of the previous understanding, right? So yet another case of you start to look into it and it turns out it's, of course, way more complicated than we thought.
Yes, exactly.
Yeah, that's kind of the motto of deep sea stuff as well. Well, it's more complicated than we thought. Oh, fantastic. Thank you so much, Antoni, for having a chat with me about your research.
And then, of course, we know that these animals are very charismatic. So one of their most endearing things to me, I think, is the fact that they have this amazing potential for science communication, right? They're great poster children for getting people excited about the ocean. They're one of these groups where you don't really need to embellish how amazing they are at all. You're just kind of a conduit for telling people about all this cool stuff that these animals just do. They're amazing and you don't need to make anything up about them to be amazing. You just have to tell people the truth. We have a couple of really impressive science communicators in our community. Someone who was talking about the way that the chromatophores function, which is Mathieu Reynard. He's been working on them for a long time and actually runs a YouTube channel called Dr. Nemo, which has some great cephalopod explainer videos on it and a variety of other interesting videos as well. So follow that link at your own peril. But of course, one of the leading cephalopod science communicators worldwide is Dr. Sarah McAnulty, who's the executive director of Skype a Scientist. Sarah gave a great workshop about cephalopods and science communication, and so I asked her to have a chat to me for the podcast as well.
Welcome, Sarah. Thanks for having a chat with me.
Yeah, thanks for having me on.
Can you tell us a little bit about things like Skype a Scientist and murals and
art nights and all of the really cool different level SciComm stuff you do?
Yeah, absolutely. So I run a nonprofit. We're called Skype a Scientist. We're based in Philadelphia in the United States. And we do a lot of different things to try to connect people with science. One of the things we do is match scientists with classrooms and scout troops and libraries, any group of people that would like to speak with a scientist. We want to lower the barriers of access to getting experts, particularly scientists, connected with people. So we offer that program for free. We serve 4,000 to 5,000 groups a year. And that program has been running since 2017. In addition to the virtual connections that we make, we also try to connect people with science offline, mostly in person in Philadelphia and a couple other cities. And so those projects are pretty art forward because we're trying to reach a population that is not already science interested. I was finding that connecting with people online was mostly attracting people who are already really into science, or at least open to science, think of themselves as science interested. And my goal is to reach people who are not in that category. So we do this using murals, street art forward night, trying to attract artists to tackle communicating on some of the big issues that humanity is facing today, like climate change, the biodiversity crisis, this sort of thing. And these projects reach people who definitely are not identifying themselves as science interested, and some of them attract a mix. So for the murals, we do community mural projects. So we really involve either neighbors associations or other community groups in the city and collaborate with them on those projects. We hire local artists to design the murals and then host community paint days to get people involved. Those murals have mostly been on the biodiversity of the area that we're making the mural in. So it's super relevant to the people in the neighborhood, and we have neighbors paint the wall with us. So they really have a vested interest in the finished work because they can say they painted it.
The scope of the different projects that you do is so cool because some of them are so wide reaching, but others are really focused on that one-on-one connection. And I think that's so important in an age where a lot of people don't understand or don't trust science. You're not sure where to find reliable information online. Having someone where you feel like they're providing reliable information and are forming that personal connection with you is also super important, especially right now.
Yeah, completely. And so part of that is why I think it's really, really critical that many scientists get involved. I think a lot of times people see scientists like, Bill Nye and these like Alie Ward, even like people who are communicating on behalf of science who reach millions and millions of people, and they might think their impact that they could have is just not worth it, not worth their time. But I think right now, particularly where we are with siloing of culture on social media and et cetera, like we really don't have time to waste not communicating and connecting with each other at the community level, at the neighbor level. Like we really need all hands on deck right now. We're out of time to lead it to other people.
And so because we've got a fair amount of early career deep sea and marine people listening to the podcast, do you have any advice for people early in their career wanting to develop their SciComm skills? And at what point can you feel that you're qualified enough to do something like Skype a Scientist or talk to other people about the cool stuff that you're learning about?
Yeah, good question. I think one nice thing about having a lot of science interested people on social media is that social media is like a good place to get your practice in. You can see what works and what doesn't on Blue Sky for sure. If you say anything that's not completely accurate or if you oversimplify, probably people are going to come in and correct you. So that for better or worse. So practicing on social media, I think, is a good start. As far as when you can sign up to be a scientist volunteer for Skype a Scientist, if you are working in a lab, you already have an undergrad degree and you're either working in a lab after your undergrad or you're pursuing graduate school, that's when we tag you in to talk with classrooms. But science communication can happen in a lot of different ways. Like I don't think that science communication right now can be limited to scientists or even professional science communicators for sure, because we need more people to engage on climate change and the biodiversity crisis immediately. So once you know what the important things for you to do are, whether it's, changing your personal approach to climate change, CO2 reductions, or like who to vote for that's going to help with this stuff, or like how you're going to plant stuff in your backyard. Like there's so many things that all of us can and need to be doing to address this problem. And so we don't have time for you to wait to get a PhD before you start talking with your neighbors about native plants or whatever. So a lot of times I think the best way to practice science communication is just doing it. And maybe you won't always get it right 100% of the time. And you shouldn't expect yourself to, because we're all learning. Just like, artists aren't built in a day. Like you're going to make bad doodles on the past. And that's probably also true for science communication.
You ran a little session on science communication at Kayak, which I thought was really great. And we had a range of different career stage people in the room. But I wanted to pick up because you were just talking about having those one-on-one conversations on a really useful pointer that you provided, right? So we talked about this thing called the empathy sandwich, which I'll get you to explain in a second. But I think particularly for deep sea stuff where there's so much misinformation and people are really fascinated by it, but there is a lot of misperceptions about it. Can you give us a synopsis of what you suggest when you're confronted with someone who has a piece of patently false information, but you still want to help them and make a connection?
Oh, yeah, this happens in so many different ways when you're communicating science. You hear people say some of the wildest stuff you've ever heard, like whether it's an AI generated piece of misinformation or fossil fuel company generated. There's lots of nonsense out there. And what you don't want to do is hit somebody that you're talking with like a well, actually, because that can really shut down the conversation. And we need to make sure people are open to listening to what we say going forward. So here's what I generally try to do. The empathy sandwich starts and ends with positive feelings and affirmations between you and the person that you're talking with. What you can start with, just find something in what they said that you can agree with. And when we're talking about deep sea stuff, maybe the thing that you agree with is like, yeah, aren't anglerfish so completely cool? And then after you have this like, yeah, this is so amazing, this thing that we both love, then you can hit them with the actual information that you need to correct. Yeah, anglerfish are so cool. But what's crazy? Like, this is actually the thing that is real. They're only about the size of a baseball or even smaller or whatever. And then you can end it with another affirmation that you both share values, you both agree with something. Like, it's so important for us to tell more people about how amazing these animals are.
Yeah, and helps them come away from that with a positive taste in their mouth and increased enthusiasm for whatever it was that you were talking about.
Exactly. Yeah.
Yeah, not feeling like they were shut down by the scientist who was smarter than you and corrected something that maybe your parent told you, so you thought it was really, really credible. And yeah.
Or even maybe they were like watching the Discovery Channel and Discovery Channel used to be reliable and now it's not, right? So you can't blame people for getting duped by a lot of nonsense. It's important that you make people feel okay when you're correcting the past. It's wild because the deep sea is so cool in reality that you really don't need to like punch it up at all. Then I see these like AI videos of monsters that are not real. And I'm like, man, all you had to do was show the real animal. Like,
yeah, what we have is so much cooler than that. Yeah, for sure. I always feel also like, especially with deep sea cephalopods, where I feel like they kind of sell themselves, right? Once you get people talking about deep sea cephalopods, you don't really have to hype them up a lot. For me, I'm just the conduit of information. Like, I know this stuff, but I'm not inventing of it anything or like creating some knowledge, sure, by like synthesizing data, but really it's all there. And if you can share it with people, like that's enough. You don't have to overemphasize anything or hyperbole it up. It's there for the knowing and it's cool enough already.
Yeah, there's no exaggeration needed when you look at Grimaldi-toothus. Like, that animal is already off the charts.
The puppet master squid, the elbow squid to ninja, there's so many. Thank you so much, Sarah. It was so great to hang out with you at Kayak.
Little bit starstruck having a chat with Sarah. That's very cool. We mentioned Scuba Scientist and the eel advent calendar on the last episode, doing some great work, always thinking outside the box, always looking at new ways of engaging with people and doing science communication in fun and engaging ways, because science isn't boring, it's amazing. We just have to sometimes talk quite dryly. And some of us forget how to talk like people after that.
That was some of her really valuable advice, how to keep people on side, even if you have to correct them gently.
But that has shown that they are seeking knowledge. You just have to be the better source of knowledge and offer them another tract.
We went diving together, we went bug spotting together, and we ate a lot of ice cream together. So I feel like that that set us up for being able to have a chat.
So that's our interviews, but I'm guessing this wasn't the only things covered in the conference. Are there any other cool bits that leapt out as topics?
Oh, so let's see. We'll summarize a 5 full-day conference in about another 2 minutes.
Yeah,
here.
we're tight
Sounds
in
good?
time, so just wrap it up.
You know, we could finish here and I'll just ring on the answer phone and summarize it in 90 seconds. How about that?
But the conference abstract is still online?
Yeah, so the conference booklet can still be downloaded, as well as the 274-page book of abstracts, including all of the authors. So if you want to know more about any of these topics or who's doing this work, that can actually be accessed by the public. So we'll have that in the show notes. I was thinking back over some of the other talks that had been really interesting. There was a lot of cool stuff about reproductive behaviors. People who know a bit about cuttlefish are probably familiar with the fact that they are one of these groups where there are different male reproductive strategies, right? So they're like the big dominant consort males who are aggressive and guarding females and sort of deterring others. And then there are the smaller sneaker males that come in, and in some, pretty extreme cases, they color themselves differently on different sides of the body so that they're displaying to the big males, I'm a female, you don't have to worry about me, while displaying on the other side to the female, I'm a male, just ignore that guy for a bit and we'll see if we can make some stuff happen. So there were some amazing talks about the fact that these strategies might be more evolutionarily locked in than we realized, because the spermatophore morphology in these two different types of male is predetermined. And so like the consort males have these really big spermatophores and the sneaker males have these smaller ones. So they're kind of predetermined to do this. It's not a matter of, oh, how big am I going to grow?
Yeah,
Which
I had a
strategy am
tough
I going to
life.
use?
I didn't grow as big as I hoped. From birth, it was going to be a sneaker.
Yeah.
Wow.
But in other species and other circumstances, there are things like the male will, we're talking about pygmy squids here. So these little tiny, they call them post-it notes of the sea because they've got a little sucker, dorsal adhesion sucker on the mantle, and they stick themselves to seagrass blades.
Ah.
And so in some of these animals, which have been studied in the lab for a long time, the male will display to the female, and if she is receptive, they will mate in a particular position. And if she's not receptive, he will do a quick flyby and just stick a packet of sperm on somewhere and head off, which she often will then remove. So she has the ability first to say, no, thank you. And then if he doesn't listen to that, she's like, yeah, I'm not storing that for use later because I said no. That's a very plastic one. The same males will do different behaviors depending on the female's receptiveness and whether they think they can get close enough.
Is the body sucker adapted from a fin, or is it a novel structure?
I believe it's a novel structure. They still have fins. This whole group has those. And yeah, they use them to maintain position attached to blades of seagrass.
Wow.
Speaking of suckers, there was some very cool talks about cephalopod suckers as well. So there are people who are in the biomimetics field who are creating hydraulic replicas of different kinds and sizes of cephalopod suckers and investigating how the strength of the sucker is influenced both by the size of the sucker and also the shape of the teeth in the sucker ring. So what kind of seal is made onto the surface based on whether you've got a few small pointy teeth or larger blunt teeth just around part of the sucker. So there were some cool talks demonstrating how they'd built these different sample suckers. There were people who had done histological work on octopus suckers and were reporting the fact that each individual sucker has multiple nerve bundles going to it, and some of those are sensory and some of those are motor. So they have completely independent nerve systems for using the suckers to grasp and release things, and for using the suckers as chemosensors and tactile sensors. So each suction cup has its own set of 2 completely different types of nerves that are controlling that sucker.
Which might explain the sort of arm sub-brain processing center.
Yes.
That's a lot of data.
Yeah. There were various discussions about, can we even agree on what consciousness is? How much of this are they controlling consciously, not consciously? How much of the camouflage is a direct automatic response to the environment, and how much of it is maybe consciously controlled? And do we call it a brain or a sub-brain in the arms? And I think the short answer on that one is no. It's a nerve processing center. They don't have 9 brains.
Even sub-brain is wrong. We'll just call it a processing center. I want to be right. I said cephalopod wrong. I want to be right.
I want to be right too, but it's not my field.
Oh, okay. Right. Bail out. Pull the cord.
Octopuses. Ugh. One other thing that was super cool actually was obviously we're interested in light and vision and how these animals are perceiving their environment. And our understanding is still that almost all cephalopods are completely colorblind, but they are sensitive to polarized light. There's some amazing work being done on what that sensitivity to polarized light might do for them. And in one case, there's a cuttlefish species that is producing a display by orienting its arms differently so that polarized light passes through the translucent tissue in a different way so that it sends one signal with most of the arms, and then it has like a little flag of a different arm that sticks up and the polarized light is refracting through it in a different way. So that this is probably a species specific sexual signal using just different orientations of polarized light.
Kat, we need to build more cameras.
I know. More cameras, more lures, more different types of light. This is a shallow water species, though. I feel fairly comfortable in the fact that at 2 kilometers, which is what we're aiming for, there's not a lot of polarized light complicating what we're trying to do. Although having just said that, I'm sure we'll be proven wrong.
We always are. Nature's amazing.
There was a leucistic octopus.
I beg your pardon?
There was a leucistic octopus. There was an individual. One single octopus, as far as I know, ever in recorded history, was brought to the attention of one of the Japanese teuthologists. He's from the UK originally, but lives in Sendai in Japan. A fisherman collected this leucistic octopus, which is all white, subsequently became named Mr. White, and was then a research subject in Ian Gleidel's lab for a while.
That's such an opportunity, actually.
So the functional outcome of this is that this octopus was basically all white all the time, was placed on different types of substrate and background to see whether it behaved as though it knew that it was not properly camouflaged. And it didn't seem to have any different behaviors from an octopus that would have typical camouflage abilities.
It was so lucky to live that long in the wild.
So lucky, right? It's like the white squirrels that you see occasionally. You're like, oh well,
delicious.
You've done very well, but I don't think you're going to be passing on a lot of your genes. He was not at all behaving as though he needed to do things differently. I think the skin texture could still be changed, but there just was no ability to color match the background.
Had no idea that it was a brilliant white.
Yes, apparently so.
Oh, that's awesome. I did have a question about the sort of mechanics of a sucker with teeth. Is it like the teeth have lips? Can you grasp a hard substrate by retracting the teeth and letting it form a seal? Or are the teeth always engaging to form a suction?
So the sucker ring sits inside a ring of muscle. And so the hard part of the sucker ring can be exposed directly to the surface or can be sort of lightly exposed to the surface with the muscle around it forming more of the seal. What tends to happen, I believe, is that the fleshy part of the sucker makes contact, the toothy sucker ring is then brought into contact with the surface, and water is pumped out of the chamber behind it to create the suction. So when the seal is complete, both the fleshy part and the toothed part are in contact with the surface. And what they're finding as they test these hydraulic replicas is that under a certain amount of strain, part of the toothy sucker ring will deform first to break the seal. And how that happens depends on the number and distribution of the teeth and how large they are, and that seems to make a big difference to both how the adhesion is initiated and how that is released at the right point in the strain.
Very, very cool. And the one little clarification I just wanted to say, I noticed one of the interviews.
GPO. GPO is giant Pacific octopus, Onteroctopus doflini, which is, I think, the gateway cephalopod for a lot of us who, have worked in aquariums or stood in front of an aquarium and just looked at an enormous octopus plastered to the glass.
A lot of people's first hug.
Yes, a lot of people. Certainly my first hug. When I worked at the aquarium in Boston, I was not actually involved in the octopus care, but of course was incredibly drawn to this animal. And so I spent as much time down hanging out with the GPO as possible. And I used to come home at the end of the day with hundreds of sucker hickeys up my entire arm up to my shoulder from the resident GPO.
Thanks so much for that, Kat. You're an excellent roaming reporter. I wouldn't have gotten all those interviews any other way. There's a real cross-section there. And because I didn't get a chance to speak directly with a lot of people who pre-recorded or used the answer phone or had a chat with Kat, thank you so much for contributing to the podcast. We are going to link to your work. I really hope that we can give you a boost as a way of saying thank you. But thank you for your time and energy. We really enjoyed covering such broad topics in the Christmas Variety Pack Delicious Chockies episode. Are you joining us for our Christmas party? For us, it's Saturday morning on the 13th.
It is in my calendar. Do we know yet what film is going to be yelled about?
We don't know what film is going to make us get very angry. It's coming together. So I'm looking forward to that. Final reminder that you can join our Patreon, join our Discord, and join us for the holiday party, which Kat has just confirmed she's going to be along to. All the cool kids will be there. We'll watch some sort of terrible film. Thanks so much, Kat. We'll catch you at the party.
April O'Neil signing off.
Hi friends, Megan here. I have an update for you on the work the Unseen Ocean Collective is doing to get ready for our show coming up in Juneau, Alaska. We are so grateful to be able to bring our deep-sea-inspired artwork and science communication outreach to the Juneau Arts and Humanities Council for the month of February 2026. In addition to madly working away on paintings, sculptures, and video installations, we are also bringing a selection of hand-drawn coloring pages for children. These pages are a collaborative effort combining line drawings of beautiful deep-sea organisms like brittle stars, mushroom corals, carnivorous sponges, deep-sea fish, and more with age-appropriate and fascinating science facts. We are hoping that this combination of art and science will help Alaska school children learn all about and connect with the underwater coral and sponge gardens in their deep ocean neighborhood. Our collective is founded on the idea that by bringing deep-sea education to underserved communities, we can foster a love for these unseen environments and nurture a new generation of deep ocean stewardship. As people, we love what we can see and connect with, which is a challenging hurdle for the deep sea since it's far away, relatively inaccessible, and in the dark. Kirsten, Nilly, Laura, and I believe that art can function as a big, bright bridge in this situation, bringing the beauty of the deep to the people, since you can't really bring the people to the deep. What could a world look like where children grow up thinking the deep is beautiful, fascinating, and engaging? How can we rewrite these alienating deep-sea narratives perpetuated by bad press and clickbait headlines? These are the questions we hope to address with our events in 2025 and 2026. We aspire to take real science about the deep sea and a real love for the deep sea in perfect combination to help inspire a new public perception. If you're keen to see our work before February, it's being shown at the Charno Marine Lab in Gothenburg, Sweden, right now until late January, and will also be in Spokane, Washington in April of 2026. You can always visit our website, unseenoceancollective.com, or follow us on Instagram and Blue Sky, both under Unseen Ocean Collective. If you can make it to any of our events in person, please say hi. We would love to meet you.
Just as a reminder, coming up very soon, 12th of December UK time, 13th of December New Zealand future time, we're still going to have our Christmassy party. So there's still time to join us.
What's the mechanism for picking the final movie we're going to watch?
There will be a vote. There will be a poll in the end. At the moment, we're gathering ideas, and then there'll be a poll.
Democracy.
Democracy in action.
Yes.
Let's see the squid do that.
Yeah.
So if you are keen to come, make sure you sign up as a Patreon pledger. And not only will you be helping to support the podcast, but you'll also get to attend the cool party. If there's ever anything I can do, I usually do. So if the podcast is ready early, they get it early. And if it was an interview that was really good, but I had to cut it for time, I've been putting up the full unedited or less edited. I'll take out the ums and ahs and bits that aren't relevant, but the longer interview I've been putting up on the Patreon as well. If there's ever a way I can say a thank you and give them something a little bit extra, that's what I've been trying to do. So we appreciate it.
Always thinking of others, Thom, aren't you? Always thinking of other people.
I'm thinking of the ones that help us keep going. Couldn't do it without them.
It's good. Someone's got to do it.
Someone's got to keep us going. There's a link in the show notes. It's on our website as well. You can just click that link and record us a little answer phone message. I think it runs for 1.5 minutes, but a lot of people just then immediately record the next part of the message anyway. Feel free to leave us a longer one as well. There's been some good stuff actually going on the Discord community, you mentioning that. Long overdue. I didn't realize how much we were missing it until we created one, but we now have an art channel. We've got so many artists that are members of the community that we now have a dedicated art channel, and some of the stuff that's been churned out is really, really lovely. Sea cucumbers, brittle star mugs, grenadier illustrations, and actual art shows that people are creating as well, and a lot more. So I can't believe we went so long without having an art channel, and it was immediately filled with absolutely amazing stuff. We got loads of feedback about the last episode. People like the bioluminescent symbiosis one. It was just nice to have such a mature field of study where all the things I was wondering about have been answered. We could really, really dive into it in so much detail. I enjoyed that one. One of our listeners has also provided a photo tour and a review of 6 amazing submersibles that they've been working with. One of the team actually won an internship, and so is getting to do some very, very cool stuff. And almost the complete opposite of that, the podcast has now pledged allegiance to Cornflake the guinea pig, and it's become a death cult, it seems. So I don't know where that went off the rails, but as soon as Cornflake the guinea pig made an appearance, people were committing their lives to this guinea pig. I didn't see that coming. I didn't see a guinea pig staging a hostile takeover.
Well, it's not the most deep sea animal I've ever heard of.
But it did come up because it's a relative of the capybara, and the capybara came up last month. Your lab spirit animal.
Yeah, that's because Javier works obsessed. Actually, he had some deep sea stickers made for this conference that has a deep sea capybara on said sticker. One of my guys, Javier Montenegro, is a really cool geneticist, and he was trying to do lots of things by, we once had this ideas from Colombia, so he said that outside America, and then capybaras been there, whatever, but he had this idea of trying to do a tracer experiment around the entire Sea of Japan, and he needed some sort of DNA that would be obviously not contamination from another part of the ocean. So he'd had this ridiculous idea of trying to completely flood the Northwest Pacific Ocean with capybara DNA.
Wow.
It never got anywhere, of course, but yeah. But then over time, it's become his sort of weird spiritual animal thing, and we've all kind of adopted it, really.
All I can think of is a giant tea bag filled with capybaras being lowered by a crane off the side of a boat and dipped in the Pacific Ocean. Dippy dippy. Well, that was sure was a podcast.
Yeah.
Yeah.
Good job, man. Good job.
Don't know if it was a good one, but it sure was one.
It definitely was recorded.
We'll do see you next time. We miss you already. We're sorry. It's Christmas, though. See you at the party.
Bye.
Would you like to reshare that story, Megan?
Sure. At our house, our 7-year-old named his stuffed alpaca toy Professor Alan Jameson because he's an alpaca, and so it's Alan Jameson. And so every alpaca toy is now Professor Alan Jameson, but they sing it, and they sing it like Hamilton. So it's Professor Alan Jameson.
I'm so glad the podcast has ruined your home.